Today, along with Dr. Hannah Gavin. It is a chilly day here in Boston, and in response to some of your requests out there, I have put my hat on. So hope it looks okay. So let's go ahead and get started with our speakers here, our session 2. And now kicking off session 2 is Dr. Ariel Furst. Dr. Furst is on the faculty at MIT. She and the members of her lab combine electrochemical methods with biomolecular and materials engineering to address challenges in human health and clean energy. For example, the Furst lab is focused on developing new technologies to combat antimicrobial resistance, detect disease, and improve microbial interactions with electrodes. I, for one, think her work is really cool, and I'm looking forward to hearing her talk. So, Dr. Furst, welcome. The screen is yours.
Thank you. Let's see if I can get this going. How does that look? Excellent. Well, first I'd like to thank Pete and Hannah and Julia for organizing such an incredible symposium. This morning's session was really interesting. So today I'd like to tell you a little bit about one project going on in our lab, which is to use DNA as a sort of Velcro to control microbial adhesion. So we like to think of this as our ability to use chemistry to help microbes interact with electrode surfaces. And our motivation for this has been, our current dependence on fossil fuels. So fossil fuels, including oil, coal, and natural gas, are a non-renewable resource. And it's known that combustion of fossil fuels is one of the largest contributors to US greenhouse gas emissions. And we can see on the right that carbon emissions since the early 1920s have increased nearly exponentially every year and continue to do so. And we know that greenhouse gases are a major contributing factor to global warming. And importantly, it's estimated that the global cost of climate change, just in terms of human health and increasing deaths and diseases, will reach $4 billion annually by 2030. So many of you might have immediately thought of our current move towards biofuels. Biofuels are fantastic, and many of them are made from things like sugar-rich crops such as corn, wheat, and sugar beets. Importantly, though, what a lot of people don't think about when we talk about biofuels are the potential drawbacks of using crop-based biofuels. This causes us to rely on things like crop monocultures, so we're no longer rotating our crops as we should to make sure the land has sufficient nutrients. Growing such crops require clean water requirements that take water away from other resources. It increases our use of chemical fertilizers. And as many of you know, this can lead to food insecurity because this land that was once used to grow food is now used to grow fuel. And similarly, this has led to an increase in deforestation, again, because we need more land to grow crops for biofuels. But what if there was a nearly infinite resource that we could use to generate electricity? Could we use wastewater as an energy source instead of crops? It's estimated that in every suburban household, 135 liters of wastewater are generated per person per day. So this is a nearly infinite resource that is continued to be renewed just by our daily lives. And one of the ways we could harness the power of this is with microbial fuel cells. So microbial fuel cells are capable of simultaneously treating wastewater and generating electricity, essentially enabling current generation from waste. And as we can see on the bottom here, we have a setup of a conventional microbial fuel cell where we have a conventional cathode that's in an aerobic chamber that reduces oxygen to water. This is separated by a proton exchange membrane from an anaerobic chamber that contains a microbe-modified electrode. And this is how we can actually clean wastewater while generating electricity. We can use the organic compounds that are found in wastewater as fuel that are converted to CO2 by these microbes in this anaerobic chamber to generate electrons. However, you might ask yourself why these aren't used widely now. It's because challenges still remain with both low energy output and consistency with bacterial growth on electrode. And one species in particular is often used as a model organism in microbial fuel cells, and this is Shewanella onadensis, or Shewanella as I'll be referring to it for the rest of the talk. And these are facultative anaerobes, which means that they can respire oxygen like you or I, but in the absence of oxygen, they can respire metal oxides. So they can reduce these insoluble metal oxides for respiration, which also means they can send electrons to an electrode surface. We can see that when they form these dense biofilms shown here, that they have these pili, and these are also known to conduct current. Importantly, these cells also have other mechanisms of transferring electrons between the cells and the electrode surface. They secrete flavins, which are small molecules that can shuttle electrons, and these cells contain cytochromes in their outer membranes that can shuttle electrons from the inside of the cell to the outside. However, there are still drawbacks to using Shewanella. To make a dense biofilm such as this one requires several days, and many of these biofilms interact with electrodes irregularly and can produce highly inconsistent currents. So we hypothesized that we could use some of the inherent properties of DNA to improve Shewanella modified electrodes. So why did we choose DNA? When many people think of DNA, they think of conveying genetic information between parents and daughter cells. But DNA has many interesting features that go beyond simply its genetic abilities. As we can see here, the structure of DNA is this regular double helix with these bases that interact through hydrogen bonding. And DNA has two key physical features that are going to allow us to improve microbial fuel cells. The first of these features is DNA's ability to self-recognize, which we've termed DNA Velcro. So with proper base pairing, we can see on the left here that DNA can hydrogen bond between itself. So thymine pairs with adenine and guanine pairs with cytosine, either with 2 hydrogen bonds or 3 hydrogen bonds. And the proper hydrogen bonding creates a double helix that's formed through a process called hybridization between the complementary strands. Importantly, this hybridization is highly sequence specific, and it's based on this proper hydrogen bonding between base pairs. Thus, we can say that complementary strands of DNA can come together and interact like Velcro does when they hydrogen bond. Importantly, when those bases come together, they form this regular helix shown here. And we can see that these bases stack on atop one another. When we look down the barrel of DNA, we can see that these aromatic bases are aligned with one another pretty perfectly. And so this double helix pattern allows the pi orbitals of those aromatic DNA bases to actually overlap vertically here. And this is known to stabilize the structure of DNA, but also facilitates the movement of electrons through this core. So we can see here that the electrons can actually flow through this core, or horizontally, we can see it flow through here. And this allows DNA to behave as a molecular wire. And this process is called DNA charge transport, which I'll talk a little bit more about later. And so we thought we could use these two inherent properties of DNA to address the two key issues we saw with microbial fuel cells. The first issue is this inconsistent attachment of biofilms to electrodes and the length of time it takes for cells to generally attach to them. Can we use DNA hybridization to actually tether the cells to the electrodes using that DNA as Velcro? And I'll go into more specifics later, but basically, if you have a surface with single-stranded DNA and cells with the complementary strand, can we get those cells to stick very rapidly to our surface? And then we sought to address this low and inconsistent current generation from biofilms. So if we have this DNA already on an electrode surface attaching our cells, can we use that inherent transport property of DNA as a wire to convey current between cells and electrode surfaces much faster than it would in a native biofilm? And so the first issue we sought to tackle was this inconsistent attachment of biofilms on electrode surfaces. And the way we tackled this is with DNA hybridization-based cell adhesion, or DNA Velcro. If we have an electrode surface modified with single-stranded DNA, we can chemically modify the surface of the cells with short complementary sequences, such that when we add those DNA-modified cells to our DNA-modified electrodes, we can get those cells to hybridize and interact with the electrode surface. And so we wanted to look at several different types of non-adherent cells beyond simply Shewanella to see if we could get these to stick to electrodes. So as we can see here on the left, we have optically transparent gold electrodes. So here's our electrode area right here. And we can see here that if we fluorescently label cells prior to their addition to the surface, we can then image those cells on our electrode. And we can see here that if we fluorescently label Jurkat cells, these are mammalian non-adherent cells, so they normally don't stick to gold. We can see if we have the proper complement on our electrode surface, we see a significant amount of fluorescence on that electrode, indicated by these white speckles. In contrast, if we have a bare electrode surface, we see little to no background binding shown here. So we looked at 4 different types of cells. Jurkat and Ramos cells are mammalian non-adherent cells, S. cerevisiae, which are baker's yeast, and Shewanella, the cells capable of electron transfer that we've been discussing. And as you can see here, not only are these cells highly specific for surfaces that are modified with DNA, but they're highly specific for surfaces modified with the proper sequence. So if we have the fully complementary strand that's shown in the solid bars, and if we have a completely scrambled sequence, so a non-complementary strand that's shown in the dashed lines. And we can see here that for all of these cell types, we see a significant amount of adhesion for the proper complementary sequence for DNA hybridization. In contrast, for that scrambled sequence, we see little to no background binding in any case. And so this showed us that our DNA Velcro is highly specific for the proper sequence. Importantly, we also found that not only was the cell adhesion dependent on the sequence of DNA, but it was dependent on the density of DNA on our underlying electrode. So shown on the left here is just a guide to show us how these different cell types vary in size. So we can see that cells range from very large, about 100 micron diameter for plant cells, down to 10 for animal cells, 5 for yeast, and 1 for bacteria. And we found that based on the size of the cells we immobilize, they immobilize to surfaces modified with different amounts of DNA very differently. So on the x-axis here is just our percent DNA. So that's how much DNA is on our electrode, ranging from 0% to 100%. And on the y-axis, we can see the relative amount of cell binding. We can see for these large mammalian cells, we see optimal binding at pretty low surface densities of DNA, only about 40%. In contrast, for these smaller S. cerevisiae, we see optimal binding at about 80%. And for the smallest cells, the Shewanella, we see optimal binding at about 90%. So what this tells us is not only can we control whether or not the cells adhere based on the specific sequence of DNA, but by varying the underlying percentage of DNA on our electrode, we can control the density of cells that adhere. And this becomes really important when we think about testing DNA charge transport to convey current between the cells and the electrode, because we want to ensure that those cells are not forming a dense biofilm, and so we can minimize the impact of other electron transfer mechanisms. So we can see here the variation in the amount of cells for Shewanella only as a function of the amount of DNA. And so for the rest of the talk, I'm going to be focusing on only using surfaces modified with 50% underlying DNA to ensure that we had sufficient cells to actually measure, but not so many that they started sensing each other and forming a biofilm. And shown on the right here, we can verify this by SEM imaging. So if we have no DNA on our electrode surface, we see no background binding of cells. If we have a surface modified with that scrambled sequence, we just verified what we saw by fluorescence and showed that we saw no cells here. And if we have that surface modified with 50% DNA, we can see here that we have a low density of cells adhered, such that if we zoom in on these cells, we can see that we're not seeing the formation of those pili that form in a dense biofilm. So now that we know that we can control the specific interaction between Shewanella and an electrode surface, we sought to address the second issue we saw in microbial fuel cells, this low and inconsistent amount of current generated from biofilms. And we wanted to see if we could use that DNA that we already used to adhere those cells as a wire to convey current between the cells and the electrode surface. So as we talked about at the beginning, the three-dimensional structure of DNA allows these bases to overlap and have their pi orbitals overlap in this regular way. And we can look down the barrel of DNA and see that it looks like a molecular wire. And we can show this ability of DNA to act as a molecular wire on an electrode surface. So if we have DNA, double-stranded, immobilized on an electrode with a distally bound redox active molecule, we will only see a signal from that molecule if electrons can flow from the surface to that redox active molecule and back to the electrode surface. And so if we look on the right here, we have this well-matched DNA sequence with this distally bound probe. And we can see by cyclic voltammetry that as we scan more negatively, we can see the reduction peak, so that peak right here, from that redox probe, and the subsequent reoxidation of that probe at the proper potential. What's really important to note about DNA charge transport is that it's highly sensitive to the proper stacking of those DNA bases. So anything such as a DNA mismatch, a lesion, or an abasic site can completely destroy the ability of that DNA to act as a wire. And we can see that with the incorporation of a single base mismatch on the right here. If we simply change that T to a C in our sequence, creating a CA mismatch, we no longer see any reduction or oxidation of that same distally bound redox probe. And so this is a really important test for us to see if a process is DNA mediated. If we incorporate a mismatch into our DNA sequence and see a decrease in our current, that indicates to us that it's a DNA mediated process. And so now we can think about looking at our cells that were immobilized with DNA as compared to conventional Shewanella biofilms. So we can see on the left here, with conventional DNA or with conventional Shewanella biofilms, we see the current grow in slowly over several days to reach its maximum. In this case, it took about 70 hours. In contrast, what we see with our DNA immobilized cells is that we see current at high levels nearly immediately after immobilization. So the process here involved allowing the cells to hybridize for half an hour on our surface, followed by measurement. And within 1/10 of an hour, so 6 minutes, we saw high levels of current that were maintained over the course of our scan. I also wanted to point out that this current is significantly higher than what we see with free cells, free Shewanella in solution, or with our background binding and buffer. But is this current that's generated DNA mediated? And as I mentioned, our test for this is by incorporating a mismatch into the DNA sequence. So here we have that well-matched DNA immobilizing our cells. We can see that in red here. But what happens if we incorporate mismatches? If we incorporate one mismatch into the sequence at different positions, we see this decrease in current immediately. If we incorporate two mismatches, we see an even larger decrease that continues to increase with 3 and 4 mismatches in our sequence. And so this indicated to us that the mechanism of our current generation was at least partially DNA mediated. But what you should be asking yourself right now is, I just showed you in the first half of this talk that if we have a scrambled sequence, the cells don't adhere. So how do we know that these differences in current are not due to different numbers of cells on our electrode surface? And we can actually measure the number of cells electrochemically using electrochemical impedance spectroscopy. And fundamentally, what we're measuring here is how permeable our electrode is to ferrocyanide and ferrocyanide. So if we just have single-stranded DNA on our surface, the ferrocyanide and ferrocyanide had reached the surface, giving us a small resistance. In contrast, once we have these large cells immobilized, that's going to block access of the ferrocyanide and ferrocyanide to our electrode and increase the resistance significantly. So we can either modify our surface with the well-matched complement to the cells, or DNA that contains one base mismatch at different locations, 2 mismatches, 3 mismatches, or 4 mismatches, as I showed in the previous experiment. And what we can see here is if we have no DNA on our electrode, we know the cells don't adhere by SEM imaging. We see this small amount of resistance indicating no cells bound. Similarly, with free cells in solution, we see no cells bound. For the fully complementary DNA, we see this large increase in resistance at our surface, indicating that we have a large amount of cells bound. And for 1 mismatch at different locations, 2 mismatches, and 3 mismatches, we see nearly equivalent cell binding to our surface. It's only when we reach 4 mismatches that we see a decrease in the number of cells bound. And we would expect this because the melting temperature of that sequence is about room temperature. So we wouldn't anticipate that those cells would adhere as well. And so now we can directly compare our current that we measured with the number of cells that we quantified electrochemically on those same electrodes. And as you see here, when we have no DNA, we have no cells bound, we see no current. If we have free cells in solution, we still see very little current, and we see no cells bound. For our fully complementary DNA, we see this large increase in both the resistance of the surface, indicating a lot of cells bound, and high levels of current. But what I want to draw your attention to is this region here, where we don't see a change in the number of cells when we incorporate different numbers of mismatches, but we do see this large decrease in the current generated at our electrodes. And as we would expect, our negative control with 4 mismatches, we see this decrease in the number of cells consistent with the decrease in current. So this really confirmed to us that the mechanism of current generation from these cell-modified electrodes is at least partially DNA-mediated. And so what we've been able to do by chemically modifying electrode surfaces and cells with complementary DNA is to use those inherent properties of DNA to improve microbial fuel cells. By using DNA as Velcro, we can use the inherent self-recognition property of DNA to actually tether Shewanella to electrodes much faster than with conventional biofilm formation. And similarly, now that we have those short strands of DNA attaching our cells to our electrode surfaces, we can use that DNA as a wire to convey current between the cells and the electrode surface. And so where do we go next? So what I showed you today was us modifying an electrode surface with a single type of cell using a single sequence of DNA. Our next step is to expand this to more complex microbial communities to modify surfaces with different types of cells and to even modify cells with one another in 3 dimensions using DNA hybridization. And this will allow us to study and control complex microbial communities to allow us to study things like host pathogen interactions, the human microbiome, complex microbial communities that occur in places like soil and ponds, and things like consortia of extremophiles. I should also point out that using DNA to attach cells to an electrode surface allows us to study unculturable cells because we don't need to be able to grow large amounts of them in the lab in order to study them. And finally, we can even think about engineering the cells themselves for producing co-cultures to make useful bioproducts or for bioproduction. And so with that, I'd like to thank the organizers again for giving me a chance to talk about our research. And I'd like to thank my group. We're a pretty small group and we're new, so we don't even have a group picture yet, but I'd like to thank my postdoc, Dr. Amrita Karvalkar, my grad students, Akiva and Aaron, and our visiting student, Manuel, and some undergrads who work at our lab, Dana and Lauren. And I also need to thank my former mentors for all of their support, my postdoc advisor, Matt Francis at Berkeley, and my PhD advisor, Jackie Barton at Caltech. And thank you again.
Thank you, Dr. Furst. That was fantastic, as always. We have time for, there we go. That's better. We have time for a few questions. So let's see. Let's start off with this one. So one of the questions is, how did you know that 50% DNA is optimal to avoid biofilm formation, but is good enough to transfer electrons to your electrode?
So the data that I didn't show there was for each of those percentages of DNA. We not only quantified them with fluorescence, but we also measured the current. And so we needed to balance basically seeing sufficient current to actually measure with the number of microbes so that we didn't see biofilm formation by SEM. And so we checked all of those at all of those different concentrations, and 50% seemed to us to be the right balance of sufficient current while not seeing those pili form.
Understood. Thank you. So our next question is, what is the intuitive mechanism that makes optimal binding scale with the surface density of DNA? In other words, how does the cell size set the scale?
So we have observed in many different sets of experiments that the amount of DNA on your surface and the spacing between those DNA strands really influences your ability to see binding or hybridization. So we've seen this with proteins that interact with DNA. We've seen this with just single-stranded DNA hybridizing, that the more spacing you have between your DNA strands allows things to interact better. So we think that with the larger cell types, you're just enabling better interactions between those single-stranded DNAs to allow them to get in the proper formation to actually hybridize. And as you decrease the cell size, you don't need as many interactions to get those cells to stick. So it's a little easier and you have a little more wiggle room.
Excellent. Thank you. So let's see. We have time for just a couple of more here. You consider two factors, DNA sequence and the concentration of DNA. What about the length of the DNA? How do you expect the results to change with length if there is any change?
That is a great question. So we have optimized our length of DNA that we used for microbial adhesion, just based on kind of balance between the melting temperature of the DNA, which needs to be sufficiently high that we can get those cells to stick, with synthetic tractability. It can be hard to make really long sequences of DNA. I can say, though, that we have observed DNA charge transport on an electrode up to 100 base pairs. So we are using about 22 base pairs for these experiments, but we do have room to expand to much longer sequences. And in fact, it's actually been necessary in some experiments with mammalian cells that have big glycocoverings to use much longer strands of DNA. But anywhere between about 20 and 100 bases is okay for DNA charge transport and should be within the realm of kind of synthetic tractability for us.
Cool. Thank you. All right. Another question for you. Would you say the connection between the DNA wire and subsequent charge-carrying cellular proteins look like? What would you say that connection looks like? Any opportunities for further hybridization?
Definitely. So the way we actually connect the DNA to the cells right now is by oxidation of surface sugars. So we're not actually connecting the DNA directly to those cytochrome-containing proteins, which is an area of future research for our lab. We know that the charge can actually tunnel over over several angstroms, basically. So we don't have to have a direct connection in order for that DNA to still act like a wire.
All right, cool. And let's ask one more here. How do you quantify the charge density when bound with DNA? So the contact area for charge to enter is no longer the entire cells over the electrode or cells per electrode, but rather the size of the DNA?
So when I was talking about charge or current density, that was based on our electrode area. So we don't really base it on the surface area of our cells. It's a pretty conventional way of measuring it, just simply based on the electrode area, but it's a really interesting question.
Yeah, for sure. Yeah, Dr. Furst, thank you. That was fantastic, as always. And I appreciate you taking the time to do this. We're going to save some of these other questions for your breakout session because you got A lot of really interesting ones here.